Hi, and welcome to my presentation on the diversity of bacteria. So for bacteria, the largest taxonomic grouping under the domain, which includes all bacteria, is the phylum. For eukaryotes, you have kingdoms under domain and then phyla, but for prokaryotes, bacteria and archaea, it just goes from domain straight to phylum. So we're going to be focusing on different bacterial phyla today, and we'll also be going over some of the other important taxonomic groupings within the phyla that we talk about. We're definitely not going to go through all of the phyla because there's quite a number of them. There's 36 different bacterial phyla that have cultured representatives that are listed in the LPSN, which is the repository for prokaryotic names with standing in nomenclature. So that would be 36 phyla that we have been able to culture species from in the lab. There's actually 10 more phyla with recognized names in the LPSN that have the candidatus status, and that's because we've not been able to culture any representatives from those phyla yet. But that's just scratching the surface of how many phyla there actually are. If you're going based on rRNA, then we currently know about 90 different phyla for bacteria. So we don't know anything much about those species within those phyla. We just know that when we collect samples from the environment, from the water, from the soil, and we analyze the rRNA for all of the prokaryotes in those samples, we find small subunit rRNA sequences that are unique and that must belong to their own phyla. So there's always more to learn when it comes to bacterial diversity. We're continuing to discover new phyla based on rRNA. We're continuing to describe phyla that we already know about by characterizing species that belong to those phyla. So every time you check, well, if you go back and check the LPSN every year, you're going to see that the number of bacterial phyla is steadily increasing. So we don't really know how many are out there, but it's estimated that there would be hundreds to thousands of different bacterial phyla in existence that we have not, most of which we have not found yet. Even though we do have 36 phyla that have cultured representatives, most of the cultured species that we have studied fall in just 4 phyla. So about 90% of all bacterial species that we have cultured and described are within these 4 phyla, which would be Proteobacteria, Actinobacteria, Firmicutes, and Bacteroidetes. So we'll go through each of those phyla in turn today and look at some of the other ones as well, starting with the Proteobacteria. So the Proteobacteria is the largest phylum, and we'll talk about it first. About 30% of all characterized bacterial species are classified in the phylum Proteobacteria, so it's very massive. All of these species are gram-negative, and as you might expect from how massive the phylum is, there's a lot of diversity in there. You have 6 different classes in the phylum Proteobacteria. So after domain comes a phylum, and then comes class next. Those 6 classes for Proteobacteria are named pretty simply. So you have Alphaproteobacteria, Betaproteobacteria, Gammaproteobacteria, Deltaproteobacteria, Epsilonproteobacteria, and Zetaproteobacteria. So those are all named just by sticking a Greek letter in front of the phylum name. Out of those, the biggest one is the Gammaproteobacteria. There's about 1,500 species that we've described that belong to that class. And then following Gammaproteobacteria, you have Alphaproteobacteria as the second largest with about 1,000 known species. And then Betaproteobacteria is the third largest with about 500 known species. The smallest would be Zetaproteobacteria. We only know one species that belongs to that class, although no doubt other species do exist, we just haven't discovered them yet. In the diagram, you can see those 6 classes for the phylum Proteobacteria. The classes are indicated by these bars to the side of the phylogenetic tree. And these names here are different genuses or genera, would be the proper plural term for genus. So these are different genera within those different classes, and they are color-coded according to important metabolic functions that they perform. So you can see there's a lot of diversity in this phylum. So we'll start with the class Alphaproteobacteria. Most bacteria in this class are aerobic, and they like to live in environments that do not have a whole lot of nutrients in them. There's 10 different orders in the class Alphaproteobacteria. The order is the taxonomic grouping that comes just under class, so it goes domain, phylum, class, order. You can always recognize when you're looking at the name of an order because it's going to end in ALES or Alice. So the biggest order in Alphaproteobacteria is Rhizobialis, which is also the most diverse order, as you might expect. There's a lot of different metabolic functions represented in that order. So you have phototrophs there, you have chemolithotrophs, nitrogen-fixing bacteria, and methylotrophs as well. The name Rhizobialis actually comes from the nitrogen fixers. Some of the nitrogen fixers in this order are the bacteria that live in root nodules on legumes, which would be bean plants. Those are called rhizobia. So they're living in a symbiotic relationship with the roots of these plants and providing them with fixed nitrogen. There's also the genus Bartonella that's part of this order, which we're mentioning because some of the species in genus Bartonella cause disease in humans, specifically Legionnaire's disease and cat scratch fever, which is an actual disease. The next order you have that we're going to talk about is Rickettsialis. These are all parasites, or at least mutualists. So the Rickettsialis bacteria, or the Rickettsias, are kind of forced to live inside of a host, which means that usually they are parasites, but sometimes they're mutualists and will provide that host with some benefit instead of just feeding off of the host. But they actually don't have the metabolic functions that they would need in order to live independently on their own, so they're forced to live inside of a host and obtain their nutrients from that host. Because of that, we say that they are obligate intracellular parasites. Obligate because they have to do it, they have to be parasites like this, or at least mutualists, they have to have a host. And intracellular because they live inside of the host cell. In the image here, you can see Rickettsia bacteria that are living inside of a host cell. So within that order, Rickettsialis, you have the genus Rickettsia, which includes bacteria that cause disease in humans. A lot of bacteria in that order cause disease in different species, but most of them don't cause disease in humans. So only some of the ones in genus Rickettsia would cause human disease. Those diseases include typhus and Rocky Mountain spotted fever. Those are both diseases that you would catch from a tick. So Rickettsia diseases generally tend to be borne by ticks or some other insect vector. And that's because they are obligate intracellular parasites. They cannot survive outside of a host cell. So they have to be transferred directly from one host to another in order to cause disease. Then you have a few more important genera that are in different orders. So you have the genus Rhodobacter and the genus Rhodospirillum. Those are both containing purple non-sulfur bacteria, so some of our phototrophs. Then you have the genus Sphingomonas, which we're mentioning because they're able to metabolize a lot of different organic molecules that are pollutants, which makes species in that genus very useful for bioremediation. And in this diagram here, you can see those different orders in class Alphaproteobacteria. This actually is not all of them. It's only kind of the biggest ones. And then some of the important genera within each order. So here you have Rickettsia in Rickettsialis. Here's your Rhodospirillum in Rhodospiralis. Here's Sphingomonas in Sphingomonidalis. Here's your Rhodobacter and Rhodobacterialis, and then your Rhizobialis with the Bartonella in there. Then we have class Betaproteobacteria, which is also very large and correspondingly diverse. You have 6 orders in that class, which we're going to go through briefly here. First order Burkholdeриalis, which is a large and diverse order. It has phototrophs in it, some chemolithotrophs as well, and some nitrogen-fixing bacteria. You have one genus in particular, genus Burkholderia, that's very important for bioremediation, similar to Sphingomonas, but Burkholderia species are able to metabolize different pollutants, including heavy metals. So when you use them together, you can get a lot of different pollutants cleaned out. Then you have the order Rhodocyclalis, which is also very diverse, which includes the genus Rhodocyclus. Rhodocyclus is more of the purple non-sulfur bacteria. Then you also have genus Zoogloia, which is useful for treating wastewater. It's going to metabolize the organic material in the wastewater, and not only metabolize it, but when it's doing that, it's going to kind of clump it together so that it precipitates out down to the bottom of the water, and that just makes it a lot easier to clean some of that organic matter out of the water. Then you have the order Neisseria, which is not as metabolically diverse. It's all chemoorganotrophs. Within there you have the genus Neisseria, which contains species that cause disease in humans. The diseases in question would be meningitis and gonorrhea. Then you have the order Hydrogenophilalis, which contains sulfur oxidizing bacteria, also some denitrifying bacteria, which would be bacteria that metabolize ammonia to some form of nitrogen gas, and some autotrophs in there as well. Then you have the order Methylophilalis, which contains methyllotrophic bacteria. No methanotrophic bacteria though, just the other types of methylotrophic bacteria. So those would be bacteria that are not using methane, but are using some other organic compound with no carbon-carbon bonds, so generally methanol. Then finally you have order Nitrosomonidalis, which includes ammonia oxidizing bacteria. So those species would participate in nitrification, oxidizing ammonia to, to nitrite. And in the picture you can see some colonies of a species in the genus Burkholderia. They have a pretty characteristic appearance. And here you have a basic phylogenetic tree of this class with all the different orders and the major genera in those orders. Next, we have class Gammaproteobacteria, which is also very large and diverse, the largest actually within the phylum Proteobacteria. You have a lot of phototrophs in the Gammaproteobacteria, especially the purple sulfur bacteria, and you have a lot of chemolithotrophs as well. There's 12 different orders in this class. We are not going to go through them all, we'll just go through some. First we'll go through order Enterobacterialis, which out of all the orders in Gammaproteobacteria is the best characterized and the best known. Bacteria that belong to order Enterobacterialis are called enteric bacteria. Enteric means of the intestines. So as you might guess from the name, these bacteria live in the intestines of different animals, but not just there, you can also find them living in the soil and the water. Some species in this order actually don't live in intestines at all. They would only live in the soil and the water, or mostly just live in soil and water, and then some of them are going to be preferring intestines and live basically exclusively there. They're all facultative aerobes and chemoorganotrophs. There's a lot of genera in that order that are important to humans because they live in our gut as part of our microbiome. So you have a lot of mutualists in there, which would be bacteria living, in your intestines as part of your microbiome and benefiting you in some way, maybe by training your immune system, maybe by helping digest your food. There's lots of different ways that your microbiome can help you in your intestines. You also have some pathogens in those genera and some pathobionts. Pathobiont is a species that is normally a mutualist, so it's normally helpful for you. But when something goes wrong or in certain circumstances, it's possible for that species to cause disease as well. So usually that would mean like if you have this species living in your intestines, then that's fine and helpful for you. But if you had some type of injury to your intestines and it was able to get out and go into your abdominal cavity, then it could start an infection and be dangerous as a pathobiont. Or if maybe your microbiome becomes kind of unbalanced, so that one species that should have just had, a moderate population level is able to have a population explosion, that could maybe cause some intestinal symptoms as well. So that would also count as a pathobiont. So just something that's normally helpful for you, but that, under certain circumstances could cause disease is a pathobiont. In this order, you also have the best characterized species of any microbe, which is Escherichia coli, or E. coli. So we know more about E. coli than we do about any other microbial species, by a good bit, actually. Yeah, E. coli is very well understood. It was one of the first to have its genome sequenced. We do a lot of work in the lab with E. coli, using it for different things. So it's a very common species to see. So we'll just go through some of the major genera in the Enterobacterialis order. So you have Salmonella, which is a genus that contains a lot of pathogens, human pathogens. Not all of them are pathogenic, but a good number of them are. In particular, they can cause typhoid fever. Then you have Escherichia. Usually those species are mutualists, so they're not harmful to you, but some of them can be pathobionts that cause UTIs if they're introduced to the vagina or the urethra. And then you also have the special strain of E. coli, the O157H7 strain, that is a pathogenic strain that causes intestinal illness, serious intestinal illness. Then you have Shigella, which is also a genus that has a lot of pathogens in it, like Salmonella does. It causes dysentery. Then you have Proteus, which is a normal species to find in your microbiome, but they frequently are pathobionts as well that would be responsible for UTIs if they're introduced to the urethra. Then you have the genus Enterobacter, which are usually mutualists and not pathobionts. You also have Klebsiella. Klebsiella, actually, those species would generally prefer to live in the soil rather than in the intestine. And they're beneficial overall because a lot of them are nitrogen fixers. But a few of them are pathogenic in humans. It's just that, normally you wouldn't be introduced to it because it's living in the soil. But if you were to be introduced to a pathogenic species from Klebsiella, it would cause pneumonia. Then you have Serratia, pathobiont. Well, you have a lot of species in that genus that are pathobionts that cause specifically nosocomial infections. A nosocomial infection is an infection that you got basically in the hospital or that is spreading in the hospital. So those tend to also be, pathogens that cause nosocomial infections commonly tend to be resistant to antibiotics as well. So you do have some antibiotic resistance in Serratia also. Then finally you have Yersinia, which contains a very serious pathogen, Yersinia pestis, which is the bacteria that was responsible for the Black Death that killed about 30% of the population of Europe. So here you have different pictures of Proteus species. Proteus species have kind of a swarming motility to them. So here you can see them in the microscope and you can see their flagella. They actually have petricious flagella, so they'd be coming out all over the whole cell body. But then when they actually spin the flagella to move, they end up all kind of twirling together into a strand. They're very fast moving. They're famous for being very fast moving. So when you plate them and grow them in the lab, it's very common to see this type of pattern forming where you have Proteus swarms moving out from a central point. And more on the Gammaproteobacteria, going through a few of the other orders now. So you have the order Pseudomonadellis, which contains aerobic chemoorganotrophs. In particular, you have the genus Pseudomonas in that order that contains human pathogens. That's another one where the species that are pathogenic are typically causing nosocomial infections that would be spread in a hospital or other healthcare setting. And especially the species Pseudomonas aeruginosa is quite common to cause nosocomial infections, and it's resistant to multiple antibiotic drugs. Then you have Vibrionalis, which contains aerobes but also some anaerobic fermenters. They're all chemoorganotrophs. Usually they live in the water, typically in the ocean. A lot of bioluminescent bacteria are in the order Vibrionalis, and they all have the shape, the Vibrio shape, which is like a boomerang, typically, although, sometimes the curve is quite subtle, but there is that curve in there. Out of the order Vibrionalis, you have one really important genus, Vibrio, which contains human pathogens. It has the pathogen that causes cholera. So here you see a Vibrio. So you can see the Vibrio shape. It's not a significant curve, but you can see the curve there. And then this is Pseudomonas. So you can see that's just a bacillus with no curve, so not a Vibrio. The next class is Deltaproteobacteria, which is also fairly large. It has 8 orders in it. A lot of bacteria in the class Deltaproteobacteria are either sulfur reducers or metal reducers of some type for their metabolism. So you have 5 out of those 8 orders in Deltaproteobacteria. You have 5 of them where the name is starting with desulf, and the species in those orders are either sulfur reducers or some other type of metal reducer, which would mean they're using that sulfur or that metal as a final electron acceptor for anaerobic respiration. A lot of the time, you'll see those bacteria living together with species in the class Epsilonproteobacteria. And that's because a lot of the species in class Epsilonproteobacteria are sulfur oxidizers or metal oxidizers. So they would be producing whatever compound the desulfo guys need to use as their final electron acceptor and then reduce it. You also have the orders Myxococcales and Delovibrionalis. Those both contain predators. We mentioned the Myxococcales in the last chapter that they have that swarming behavior, and then they form a fruiting body and distribute spores after consuming a meal. The Delovibrionalis don't do social hunting, but they're also hunters or predators. And then you have order Syntrophobacterialis, which contains syntrophs. So those would be bacteria that live in a symbiotic relationship where they're working together with another species to metabolize something. These syntrophs in particular are producing molecular hydrogen as a result of their metabolism. And they need to live with other species that are able to consume that hydrogen as they make it. Because if the hydrogen is not consumed, then the reaction is not favorable for these Syntrophobacterialis species, and they won't be able to kind of make the reaction go. Then we have the Epsilonproteobacteria, which is much smaller with just one known order, the Campylobacterialis. You have a lot of sulfur oxidizers in that order, which would frequently be found living together with species in those 5 orders of Deltaproteobacteria that start with disulf. You have a lot of spirilla-shaped bacteria in the Campylobacterialis, so those are bacteria that have kind of a loose spiral to their shape. And you have a couple of important genera for humans. So first, the genus Campylobacter. Species in that genus frequently cause foodborne illness, and they're kind of the major cause of foodborne illness today, in the US anyways. Then you have the genus Helicobacter that also contains human pathogens. Specifically, you have H. pylori in there, or in that genus, which is an acidophile that is able to live happily in your stomach and causes stomach ulcers. Also, other things can even cause cancer in some cases. So Epsilonproteobacteria is probably larger than what we know of it right now, just one order. And actually a lot of the species that we do know of that are in this class, we haven't actually characterized them or cultured them or anything. We know them only based on their rRNA sequences. So there's a lot of diversity to uncover in the Epsilonproteobacteria. So that was our major Proteobacteria phylum. Now we'll go through some other phyla. So we have the Firmicutes, Deinococcus, and Actinobacteria up next. Those are all grouped together because they're all gram-positive and they're kind of closely related to each other. So all in all, they're quite large, especially the Firmicutes and Actinobacter are large phyla. The 3 of them together have about half of all known bacterial species, so they're quite significant. The Firmicutes and the Deinococcus are all low GC content gram-positive bacteria, which means that if you sequence their DNA, you don't find a lot of GC pairs. You would find a lot more AT pairs than GC pairs. Then Actinobacteria are high GC content bacteria, which means they do have a lot of GC pairs in their DNA if you sequence it, and not as many AT pairs. So we'll start with the Firmicutes, which is a large phylum, has 7 classes in it, a lot of different orders, but 3 of them that we will go over. So first, order Lactobacillis. Members of that order are called lactic acid bacteria because of their metabolism. They do lactic acid fermentation. They're very important for food production. So there's a lot of different species of bacteria in different taxonomic groups that are important for food production, but probably out of all of them, the lactic acid bacteria have to be the most important, or kind of the greatest concentration of important species for food production. So your dairy products, milk and yogurt and cheese, different cheeses, pickles and different fermented products are all produced using these lactic acid bacteria. They aren't just fermenters, they are also aerotolerant. So they're able to tolerate, an atmosphere that has oxygen in it, even though they don't use that oxygen for respiration. And they always need sugars to kind of feed off of in order to do the lactic acid fermentation. A couple of important genera in this order would be genus Lactobacillus and genus Streptococcus. Lactobacillus has acidophiles. They're not pathogenic. A lot of them are mutualists, so it's very common to find Lactobacillus species in your microbiome, including in the vagina. And a lot of probiotics will contain Lactobacillus species. In general, it's considered, healthy to consume them. Then you have the genus Streptococcus, which is a bit less healthy as it contains some human pathogens, in particular the species that causes strep throat. And in the image here you have different views of species in Lactobacillus. So you can see they are, bacilli. They form these long chains. This one is in the middle of dividing by binary fission. Then we have the phylum Deinococcus, which is a smaller phylum, or at least a not very well-known phylum. You only have one class in that phylum right now, which is the Mollicutes. Bacteria that are in that class or in that phylum are called mycoplasmas, after this one genus in that class, which is Mycoplasma. So you have within the class Mollicutes, you have the genus Mycoplasma, but also any bacterium that is in the class Mollicutes will be called a mycoplasma, even if it's not in the genus Mycoplasma. That's because all of these bacteria are having features in common that are pretty striking. So specifically, they don't have cell walls, like essentially all bacteria have cell walls. So it's very unusual that the Deinococcus bacteria do not. So that creates some problems for them that most bacteria don't really have to deal with. The average bacteria with its cell wall doesn't have to worry about being in a hypotonic solution, but a mycoplasma bacteria would need to worry about being in a hypotonic solution. Without the cell wall there to protect it, you can have water rushing into the cell and causing it to swell up and pop or lyse. So in order to prevent that from happening, they have kind of unusually strong cell membranes that have a lot of sterols and lipoglycans in them. Sterol is a type of lipid related to cholesterol. So, animal cells have cholesterol in their cell membranes to help stabilize them, but bacterial cells do not in general, with the exception of these mycoplasmas. It's a quite unusual feature, and also a few other bacteria that have sterols as well, but it's very uncommon. And a lipoglycan is just a polysaccharide that has, some lipid attached to it. I'm sorry if you guys hear some of that rustling. It's my cat. He will not stop. Having these sterols and lipoglycans in the cell membrane for the mycoplasmas makes the membrane stronger, which helps it resist lysing. But it's not enough in general. So most mycoplasmas are going to be intracellular bacteria that live inside of a host cell, and then the host cell is maintaining an isotonic environment for them, where the same amount of solutes that are inside of the cell, you also have the same amount outside of the cell. So in that case, water isn't going to be kind of moving across the cell membrane by osmosis one way or the other. Also, as a result of these mycoplasmas not having a cell wall, they have pretty variable shapes. So you can have cells of the same species that have kind of strikingly different shapes. The shape of the cell can change over time. And a lot of them are pleomorphic, which means that they don't have really an identifiable shape. So here you have some colonies of mycoplasma, and here's a kind of a closer view of what they actually look like. So you can see the really strange, amorphous, almost shapes that they have. So next you have the phylum Actinobacteria, which is our last gram-positive phylum. It's also a very large phylum with 6 classes in it and 9 orders. I should say actually 9 orders that are kind of considered important. It actually has more than 9. But out of those 9, we're actually only going to talk about one, which is the order Actinomycetalis. You have a number of genera in that order that are important to us. So first we have genus Chlamydiae, which is just interesting because it has a very unusual type of cell division called snapping division, where kind of right before the cell is going to finish dividing, the cell wall has to snap. And that causes the arrangements that you see in the Chlamydiae, which would be the V-shape arrangement and the palisade arrangements. So you can see in the picture here, those Chlamydiae, when they divide by snapping division, the cell wall of those 2 cells remains attached kind of on one side, and then on the other side it snaps, and that makes the cells kind of swing to form this angle with each other. which we call the V shape. So these are all V shapes, except for this one is almost more like a palisade. In the palisade, the cells would actually be kind of parallel to each other, but you wouldn't generally have just two cells, you would have kind of a few of them in a row. So if this guy kind of continues dividing, probably that's going to form a palisade. Then, in addition to the Corynebacteria, you have genus Propionibacterium. So those are anaerobes. They like to eat oils and fats. You have some of them living on your skin, actually. I won't say that they're part of your microbiome necessarily because of the implication that that means, that it's good. But Propionibacterium, they can't live on the surface of your skin because there's too much air there. They need to live down in your pores where it's an anoxic environment for them. And then you have your oils down there that are produced by your skin. And so they're able, to hide from the oxygen down there and eat the oils. And then your immune system finds them living down there and responds to it. And then that causes the formation of a pimple. So these guys are responsible for acne, even though technically it's your immune system's response that is kind of making the pimple. So if your immune system did not respond, you wouldn't have acne. There's also a species in that genus that's important for cheese production, specifically for making Swiss cheese. Then you have the genus Mycobacterium, which has really unusual cell walls. They're very waxy and thick. Some members of that genus also will grow super slowly. It can take them, kind of weeks to months to divide, which is very unusual for bacteria. You have some human pathogens in that genus. Specifically, the species that cause tuberculosis and leprosy are Mycobacterium species. So you can see that right here. So this is a colony of a Mycobacterium species. And it has this strange appearance because of the waxiness of the cell walls. Then finally you have the genus Streptomyces, which has an unusual shape to it. The species in that genus are going to form hyphae, which you can see in the image. These strands are the hyphae. But the genus Streptomyces is really important for us because it is the single largest source of antibiotic drugs that we have. So it's quite common to get antibiotic drugs from nature, from some microbe, a bacteria or a fungus, even a plant sometimes. And then you would usually modify the compound that you got from that natural organism to increase its effectiveness and decrease its toxicity and stuff like that. So the Streptomyces genus has been the source for more antibiotics than any other natural source. So it's very important medically for that reason. So then we have some other phyla that we'll go through. One of them is the Bacteroidetes, so that's a very large phylum, and the rest are just kind of some of the small, more miscellaneous phyla that have something important or interesting about them. So first, the Bacteroidetes, a large phylum, as I mentioned, one of those big four largest phyla. So you have over 700 species that are described so far in this phylum. So it's very diverse. It has 7 classes and 7 orders that are kind of considered more important. It's gram-negative. You have a lot of bacteria in this phylum that are able to kind of digest cellulose and other complicated polysaccharides like that, which is kind of an uncommon ability. Usually those really long structural polysaccharides are really hard to digest, which is why they're so durable. So the Bacteroidetes are unusual in being able to digest those things. And they can do that for you as well, since a lot of them will live in your gut as part of your microbiome. And one of the things that they are doing for you is digesting polysaccharides that are in your diet so that they're kind of chopped up into smaller bits that you actually can digest, because you yourself cannot digest polysaccharides. at least not the structural polysaccharides. So you have order Bacteroidetes in this phylum. That's including anaerobes, basically. Most of them are fermenters, and most of them are obligate anaerobes, so they cannot ever be exposed to oxygen. This is the order that has the microbiome members that I mentioned, specifically the genus Bacteroidetes. Species in that genus will like to live in intestines of some type of animal. A lot of them live in human intestines. In general, they are either commensals or mutualists. So either as a mutualist, they are helping you in some way, or as a commensal, they're not doing anything to help or harm you regardless. They're just kind of there, not hurting you, not helping you, just a neutral effect. Some of these can also be pathobionts, though, if they do escape the intestines. That's usually not such a problem because the intestines are an anoxic environment, but if they were to leave the intestines and go, say, into your abdominal cavity, you do have oxygen there since all of your cells require oxygen to live. Oxygen is being distributed all throughout your body, and an obligate anaerobe cannot survive there. So in general, if they escape the intestine, they will die before they can cause disease. But there are some exceptions. There's some situations in which they can still establish a foothold and cause an infection. Then you have the order Flavobacterialis, which members of that order are common to find living in water, especially in cold water, so kind of in the oceans closer to the poles. And as you might expect with that habitat, a lot of them are either psychrophiles or at least psychrotolerants. And then this image here is different Bacteroides species in the gut of an animal, and they're kind of color-coded. So in blue you have one species of Bacteroides, green would be a different species, red yet another species, yellow yet a fourth species, and then this purple stuff is food. It's not microbes. Next we have the phylum Chlamydiae, which is a pretty small phylum. It only has one class and one order. And the order is Chlamydialis. The species in this phylum are all gram-negative, and they're obligate intracellular parasites, just like the species in order Rickettsialis, which would be back in phylum Proteobacteria. So this Chlamydiae phylum and that order Rickettsialis, those are kind of the biggest examples of obligate intracellular parasitic bacteria that we have, where they're organisms that don't just live as parasites inside of a host cell, but they have to. There's no way for them to survive otherwise because they've lost a lot of the metabolic capabilities that they would need to survive on their own without a host. The species in phylum Chlamydiae have really small genomes. So, of course, all bacteria in general would have small genomes, but these are really, even extra small, they're really, really, really small. Obligate intracellular parasites actually generally do have small genomes because of all those functions they've lost. They've lost those functions because they've lost the genes that would allow them, to make the proteins that they need for those functions. So they have a very restricted genome. Everything they need that they don't have, they're getting from the host cell. You actually do have some eukaryotic genes in the genome for certain Chlamydiae species, which is very strange. And we think that has been gained over time from host cells by horizontal gene transfer. You have two different life stages for bacteria in the phylum Chlamydiae. Whenever the bacterium is between hosts, it's going to form an elementary body, which is kind of hardy. It's more like a spore. It's not an endospore, but it is more resistant to harsh environmental conditions than a normal cell, and it's kind of dormant. So it's just traveling from one host cell to the next in that stage. Then when it gets inside of its next host cell, it's going to change into a reticulate body, which is the vegetative cell, or the kind of the normal form of the cell. So it's metabolically active. It's going to start, metabolizing in the host cell, reproducing there, and then those offspring will at some point form into an elementary body and then be released to find a new host cell. So actually you can see that cycle here where you have elementary bodies outside of a host cell, then they're going to get inside of their host cell. Then they convert to a reticulate body, so you can see that's kind of larger. The reticulate bodies are going to be multiplying inside of the host cell, and then once a lot of them have multiplied, some of them will start converting to be elementary bodies, and then those elementary bodies will be released. Generally, when that happens, the host cell will die. So most of these species are going to be causing disease in some type of host, but in the genus Chlamydia, you have species that are specifically infecting humans and causing the diseases chlamydia and trachoma. Trachoma is an eye disease that you might not have heard of, but it actually is very common worldwide, where basically you have these bacteria infecting your eyelid, which makes the surface of the eyelid rough, and then when you're blinking, it drags across your eye and damages the cornea and can cause blindness. In this image here, you have a host cell that's being infected with chlamydia. And so you can see all these kind of black dots are the elementary bodies. So there's a lot of different elementary bodies in this host cell. And you can actually see that it's kind of getting a little fuzzy on this end. So my guess is they're going to start coming out over here. Then we have the phylum Planctomycetes. These are also gram-negative bacteria, and we're mentioning them because of their morphology or their shape, which is quite strange. So they will usually have some type of prosthecae, maybe stalks, maybe appendages, maybe something else, and usually they use budding division instead of binary fission. A lot of them also have intracellular membrane folds, where they would be doing some form, something for their metabolism. What they're doing specifically with those folds is going to depend on the species and what type of metabolism that species is using. Some of them also have an S layer in their cell wall, which normally is something that you only see in archaea, so it's very strange. Maybe it's there because of horizontal gene transfer, but it's not really clear. This isn't a very large phylum. It only has 2 classes in it and 3 orders. A couple of those structures that are unusual that you find in the Planctomycetes are the parallelosome and the anamoxisome. Actually, most Planctomycetes don't have the anamoxisome. They would only have that if they're doing, if they're ammonia oxidizers. But every Planctomycete has the parallelosome. So both of those are structures inside the cell that are formed by membrane folds. In the parallelosome, you have the nucleoid inside of it and also the ribosomes inside of it. So it's almost like a protonucleus. So it's really interesting. I guess one of the main things that separates it from being a nucleus is just that the membrane around it doesn't have 2 layers, it's just the 1 layer, and it's actually attached to the cell membrane, so it's not like a separate membrane. Then the anamoxisome is for the ammonia oxidizers who are participating in nitrification. So they're going to be doing ammonia oxidation inside of there, which has some pretty toxic intermediates. So that's the reason why they need kind of a special compartment to stick the enzymes for that process. In this image here, you can see the parallelosome of a Planctomycete. So you can't see it here, but this membrane is actually attached to the cell membrane. It would just attach kind of in a different plane, so maybe up from this image or down beneath this image. Then you see this kind of hazy region inside of the parallelosome, which is the nucleoid, and you would also have a bunch of ribosomes floating in here. And then in this phylogenetic tree, you can see that the Planctomycetes are actually not that far from the Chlamydiae in terms of their relationships. They're pretty closely related to each other, actually. Then we'll go through a few of the Thermophilic phyla. This is not all of them, and of course it's certainly not all thermophilic species, which are distributed in a lot of different phyla. But for these three major thermophilic phyla, all of the species in the phyla are thermophiles or hyperthermophiles. So first you have Thermotogae. Actually, all of these are gram-negative, but the Thermotogae have sheaths. They are anaerobes, so they do not tolerate oxygen. They reduce iron as their final electron acceptor for anaerobic respiration. They actually picked up the genes they needed to adapt to the hyperthermophilic lifestyle from archaea by horizontal gene transfer. So those are actually archaeal genes that they're using to survive the high temperatures. You can see them on the side here. This is the sheath, this kind of thin gray line. So in black you have the actual bacterium, and then the sheath is around it. Then you have Thermodisulfobacteria, also gram-negative, also anaerobic, not hyperthermophilic, just thermophilic, and these are sulfate reducers, so using sulfate as their final electron acceptor for anaerobic respiration. They also have taken something from archaea, it seems, since some of their membrane lipids are very similar to lipids that you would find in archaea. They're not like the normal lipids that you find in bacteria. And it's not clear why that is either. Then you have the phylum Aquificae, which is also gram-negative thermophiles. They have a more diverse set of metabolic options to them, or I guess different species have different metabolisms that they're using. You have a couple important genera in that phylum. First, genus Aquifex. You have chemolithoautotrophs in that genus, so not only are they getting their energy from inorganic sources, but they're also getting their carbon from inorganic sources. They're important or unusual because out of all bacteria that have been described, the most hyperthermophilic bacteria that have been found are in the genus Aquifex. A lot of them are sulfur oxidizers, so they would be basically taking their energy from sulfur, oxidizing it as an electron donor for respiration. Here's an Aquifex bacterium off to the side. Then you have the genus Thermocrinus, which is just important historically because the first thermophilic bacterium ever described was in that genus. And you can see that up top. So the Thermocrinus is actually right here. This is kind of runoff from a hot spring, and the Thermocrinus bacteria form these long filamentous streamers that are kind of red-brown in the water. Actually, sometimes they look pink. Then we'll talk about the phylum Deinococcus Thermus, which is unusual because it has some UV-resistant bacteria in it. So you have one class in this phylum and two orders, so it's not very large, or I should say it's not very well known. Undoubtedly, there's plenty of other species out there that would fit into this phylum if we could just find them and culture them. The species in this phylum have a really strange cell wall. They're not really gram-negative or gram-positive. They're mostly like gram-negative bacteria in their cell wall, except that they don't have LPS. So you can actually see a close-up of their cell wall right here. So this would be the cell membrane. Then you have this kind of dark layer that is their thin layers of peptidoglycan. Then they would have the periplasmic space, and then on top of that, the outer membrane, which you can see here. So normally for gram-negative bacteria, you would have LPS embedded in the outer membrane and sticking out, like you would have long tails of it sticking out above the membrane. And that LPS ends up being pretty important because it keeps things out of the membrane. It makes the outer membrane not as permeable as a normal membrane is. It makes it so a lot of things that can get through a regular membrane cannot get through that outer membrane. Of course, that doesn't apply to the Deinococcus Thermus species since they do not have LPS in their cell wall. So they actually stain gram-positive, even though their cell wall looks more like a gram-negative cell wall. They would stain pink, like red or pink, like a gram-positive bacterium. So you have to kind of identify them otherwise to make sure you know like what they are. You have, or I guess the two orders that you have in this phylum are Thermalis and Deinococcales. In the Thermalis, as you might guess, they are thermophiles for the most part. The Deinococcales are really interesting though, because they're resistant to radiation, like UV radiation. So UV radiation damages DNA. One of the ways that they have gained resistance to radiation damage is that they just have really strong DNA repair mechanisms. So they really repair their DNA very quickly and very well. So when you have the radiation come in and it damages the DNA, they just fix the damage. So that means that not only are they resistant to radiation, but they're also resistant to other things that cause DNA damage, like certain chemicals that cause DNA damage. So they're really interesting, and seems like maybe they might have some secrets for us, perhaps. One unusual thing about them is that their nucleoid has this weird organization, or it looks like a donut. This is actually called a torus, this shape, a circle with a hole in the middle. That's a torus. So we'll conclude by just going over a few of the phyla that we know only from their rRNA sequences. So for all of these, we don't have a single species that has been cultured and grown in the lab and really studied. So what we know about them is really limited. We do know a bit more than just their rRNA sequences, because we get information from like, where did we get that sample from? Well, that's where that bacteria lives. And we can also sequence other parts of the genome, not just the rRNA, to get information about what type of proteins they have, which tells us like what type of metabolism they probably use and stuff like that. So we know a little bit about them, but just really not very much. And we have no idea about or very little idea about kind of the diversity that's in these phyla. So first we have Acidobacteria, which we actually mentioned in the previous chapter in reference to photosynthesis, because there's been a phototrophic species discovered in this phylum. That's also acidophilic. Then there's also the Gammaproteobacteria phylum, where a single phototrophic species was also discovered. Then you have the phylum Nitrospirae. These are nitrite oxidizers and autotrophs, so they're participating in nitrification. We had talked about ammonia oxidizers before in this chapter. So one half of nitrification is ammonia oxidation, oxidizing ammonia to nitrite. The other half of nitrification is nitrite oxidation, oxidizing nitrite to nitrate. And that's what these guys are doing. They're also autotrophs, so they're getting their carbon from some inorganic source. What's unusual about the Nitrospirae members, some of them, is that some of them seem to have the enzymes they would need to do the nitrite oxidation, but also to do the ammonia oxidation. So usually species can only do one or the other, but you have some species in that Nitrospirae phylum that seem that they can do both. Then you have the Synergistetes, the Fusobacteria, and the Fibrobacterias. So those are all gram-negatives. They're all anaerobes. They all use fermentation for their metabolism. And we know about them because they're in our microbiome or in the microbiome of other animals, either in the intestines or on different mucous membranes where oxygen is not commonly present. So it'd probably be good to know more about them. It would be good to learn more about them since they're inside of us.